All right, welcome back. So now we're going to move on to the next part, which is translation. So we talked about how we copy the DNA through replication. The last lecture talked about how we went through the process of transcription, going from DNA to RNA. Now the last step is translation, when we take the RNA and code it into an actual protein. Now this is going to happen in the cytoplasm. So remember, transcription and replication had to happen in the nucleus because you're using the DNA as a template. Now the RNA, the messenger RNA, is able to leave the nucleus outside one of those nuclear pores, and it's going to go to where it will find a ribosome. The ribosomes can be either free-floating or attached to the endoplasmic reticulum of the rough ER. Now we're going to need a few things. We're going to need that messenger RNA that we made in the previous step of transcription. We're going to need the amino acids, which are your building blocks for proteins. It's going to take some energy, and we're going to need some helper molecules. We're going to need some ribosomes and transfer RNA, and both of these components are actually made of RNA. So how do we go from a series of letters to actual protein? So if you remember the proteins they coded for, I gave you like a little 3-letter word for them, or sometimes it has a 1 letter. There were 20 essential amino acids, and each of them relates to a specific code. Now, what's going to happen is your cell is going to read your transcript of the RNA, and it's going to break it down into parts. And how it's going to do, it's going to break them up into these little 3 parts of 3 called codons. So each codon is going to specifically code for a specific amino acid. So this combination of AUG codes for methionine. ACG will code for threonine, and so on. And your cell will go through and use this code and what each corresponding codon relates to its corresponding amino acid. Now, depending on where you start, this entire thing right here is called your reading frame. And you'll see we'll have things called start codons and stop codons to tell us where to start and where to begin. So let's look at the reading frame, because where you start really does impact what your protein's going to look like. So remember, each codon codes for a specific amino acid. And if you just change it by 1 letter, it's going to completely change the sequence. So here's our frame right here. Here's our messenger RNA. So what we're going to do, we're going to start the reading frame right here at the start right here. So here's our first codon, second codon, and so on. So if we use the reading frame where we start right here at the end of the 5' end, our first protein, our first amino acids is serine, and the next one codes for a stop codon, and then we have another serine, arginine, lysine. All right, so this would have been, part of a protein, then maybe starting another one. Now what happens if we shift it over just 1 base? So now our first codon is CAU, and this is the amino acid chain that we get. So we have a histine, asparagine, leucine, valine, arginine, so on. Now let's just shift it over 1 more. Now we start with the methionine, alanine, serine, stop. So just by moving it over 1 base, you got 3 totally different proteins. So how do you know what combination correlates to which amino acid? Well, we have this genetic code, and this genetic code is universal. Regardless if you're a bacteria or a eukaryote, you're going to use the same genetic code, and that's one of the reasons that we support that we have this common universal ancestor. Now, there's 64 combinations, and how you read this is you have your first letter here, second letter here, and third letter here, and this will help you kind of go through and map on the table. Now there's a few things. There's one thing called our start codon. Every protein's going to have methionine as a start codon. It's one of the key things that we have to have. We're also going to have 3 stop codons, and these are going to be important for termination of translation. Now another thing I want to point out is you see on some amino acids multiple codons, code for the same amino acid. This redundancy in the genetic code kind of helps along with sometimes when we get mutations and something called the wobble effect. So you'll use this a little bit later on, and in one of your assignments you had to go from DNA to RNA and RNA to protein. As I mentioned, we need some helpers with this one. We're going to need a transfer RNA and a ribosome. Now what tRNAs do is they bring the amino acid that relates to a specific codon to the growing protein chain. So there's different ways that we show tRNA. It's just a long strand of RNA, and then it folds in on itself. It can look like this shape right here, or this T shape that we're used to seeing right here. Now, on part of the tRNA, we have what's called an anticodon loop. This anticodon loop matches with the codon, so it's going to have the corresponding bases. And this is going to have an amino acid that's going to be, boom, that's going to be charged right here. So if a tRNA molecule doesn't have an amino acid attached, it's uncharged. When it has one, it's said to be charged, and what it does is it's going to go along here and it's going to scan till it finds a matching codon that matches with its anticodon. And it's going to bring this amino acid to the ribosome, and that's going to help us build our protein chain. Now the ribosome is pretty much your protein factory, if you want to think of it. It's going to be composed of two subunits. There's a bottom subunit and a top one, and it's going to serve as a site to where we can make those peptide bonds and grow our protein chain. Now there's three sites that you'll see. We're going to have an exit site, an A site, and a peptidyl site. So you're going to arrive site, exit right here, peptidyl site where we're going to grow that peptide bond, and then an exit site. So basically what's going to happen is just serves as a site for the tRNAs to come, bind to the messenger RNA, and then what we're going to do is transfer the amino acid to a growing chain to make our protein. So just like with replication and transcription, we got to start it, we got to grow it, we got to stop it. So let's talk about how we start this. So we need to make sure we have our ribosome. So we're going to have a subunit and a larger subunit. They're not going to be joined together at first. Eventually, we're going to have the small subunit by itself, and we're going to have to kind of build this little sandwich in a way. Now what's going to happen is the small subunit is going to go through and scan along looking for a certain sequence, which is that start sequence. Now, this lower subunit is going to be loaded with the first tRNA that codes for methionine. It's our initiator tRNA. It's always going to be it's always going to be the first one. Now, what it does is it hops onto the mRNA strand at the 5' end, and it just scans. Scan, 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 scan. It is looking for the sequence right here, AUG. Now, once it finds AUG, it stops. And then that stopping signals this larger subunit to come in and bind. Now, when it binds, this initiator is going to be in the peptidyl site, or that P site. So initiation is having this small subunit with initiator scan the strand until it gets to AUG. That's going to stop it, and then that will allow the top subunit to go. So bottom bun, top bun, there's the meat, if you want to think of it that way. Now, elongation, we're going to focus on this one down here. So what's going to happen now is once we have that initiated one starting, we have this empty amino site, all right? So what's going to happen is the next tRNA that matches up is going to come into right here. So this anticodon is looking for this codon. It's going to come in, boom, 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 boom. And once it matches up, it's going to start to do a few things. Now, once you're here, you're not going to keep, these tRNAs do not keep their amino acid. They want to get rid of it so they can go get recharged and do that. So what happens, once the new one comes in that matches, it's going to, the one that's here in the P site is going to transfer its amino acid to the next one. So what happened here was tRNA came in here and matched, coded for arginine, and what happened was this tRNA transferred its amino acid to that one. And we're going to start to grow a chain. Same thing happened here. This amino acid was transferred to here. Now, once you have no amino acid attached, you kind of slide down and you go to the exit site and you leave. So this process is going to keep going in this direction. There's some good videos at the end that I advise you to watch because it's really confusing at first. But just a matter of matching up, coming to the side, and transferring your amino acid to the next one. And then termination is quite simple. I think it's one of the easiest ones. Remember, we have those stop codons. Now we're going to keep going and scanning through and adding the appropriate amino acids right here until we get to a stop codon. Now the stop codon doesn't actually code for an amino acid. It codes for some what we call release factors. Now what happens is when a stop codon is reached, it matches up with there, the release factor binds. And when the release factor binds, he pretty much ends the party. He's like, nope, party's over. What that does is it causes the protein to be released, and then all the subunits come apart. So now we are able to reassemble this and make another protein. So this machinery is reusable over and over again. The mRNA probably has a limited life depending on how it's being degraded. So termination is easy. You just find a stop codon and it's kind of the buzzkill and breaks up the party. Now you're going to be asked to do this on one of your homework assignments. It is actually go through the process of transcription and translation. Now you did transcription on the previous one. Now translation, this is just showing you how to use the table. So what you're going to do is, here's your codon right here, and you're going to match it to the table. So use these letters to help you find it a little bit easier. So our first one is AUG. So here's A, and I bring down the U, and I look for the G. And that codes for, we type it in, met. All right, let's do the next one. ACU. So A, go for the C, U. And that one will code for, I gotta switch between typing that, THR. And you just keep going on until you get to the stop codon, and you can write the word stop. But if this is giving you trouble, please let me know. And then this is just giving you a little bit more practice if you want to do that. So you would do the same thing where you went from DNA to RNA, and then RNA to the amino acid. And remember with the RNA, remember there's no T's. That's why you don't see any T's in this table. So if you have a bunch of T's here, change them to U's. So I wanted to keep this one kind of short. These help a lot. Watch them because I know transcription and translation are really hard, and we would have spent quite a bit of time in lab and lecture going over it. So don't be afraid to ask for help, and then I will see you next time.